Okay, good morning, everybody. Just have to use my computer microphone today, so I hope that you can hear me all right. And today I'm going to finish off talking about the eye. We talked about this briefly last Wednesday when we also had the review session. So we have a short lecture to start this discussion of the eye off, and then we'll complete today. Please, there's a couple of things I want to say about test taking. Please, can you all check your calendar really carefully to make sure that you know when the dates of the tests are. Second, can you all please check your email addresses? The email address that you have registered with St. Francis College, that is the email address which I'm using to communicate with you all. If there's anything wrong with that email address, you're not going to be receiving notifications. Thirdly, as far as the tests themselves are concerned, if there's any problem with the time that you take the tests, it's as stated on the syllabus, all tests are taken during the class period, during the posted class period. That's 10 past 10. I actually open the tests usually at 10 o'clock. So they go for the whole period of the class. If there is a problem with that time for any particular reason, it must be a good reason, then I need to know about that beforehand, not afterwards. You cannot contact me after the test is finished and say, oh, I couldn't do the test. Can I do it now? These things, unless there was a dire emergency, these things need to be arranged with me beforehand. First of all, for the purposes of security, the whole class should be writing the test at the same time. Second of all, in terms of fairness, people who want to write a test later on, I even get people writing to me after a test and saying, I didn't take the test because I wasn't ready to take it. If you're taking a test later than the rest of the class, you've got more time to study than the rest of the class. And that is simply not fair. It's not fair to those who do set aside the time to finish the test during the set time. Okay, so please, for the upcoming tests, please be aware of that. You have one more test during the semester, and then you have a test at the end, on the day of the final. One half of your final test, is composed of a normal class test. The other half of the test at your final is going to be a comprehensive, which is going to be material that you've already been tested on. So just make sure that you know those dates and that you've made arrangements with work, whatever, that you have that time during the class period in order to complete the test. Okay, so bureaucracy aside, let's just have a look at what we talked about last time. And this was, we discussed briefly the structure of the eye, and I'm just going to run over it once more because this is really important. So first of all, we learn that the eyeball is made up of three important layers. The first is this layer on the outside, which is a thick, fibrous white layer, which we see as the whites of the eyes in human beings, the whites of the eyes between when the eyelids are open. And that is called the sclera. And the sclera is opaque white until it reaches the very front of the eye here, where it's completely clear in order that light can pass through, obviously, into the eye. And that is this clear, absolutely clear tissue with no blood vessels in it is called the cornea. The whole thing is covered with a thin layer of epithelium, which is called the conjunctiva, and richly invested with nerve endings, blood vessels, and all the rest of it, except over the cornea, where it does have nerve endings, but no blood vessels, because they would obscure the image, the light, interfere with an image formation. So behind, on the next layer of the eye is here. It's a thin layer, called the choroid. There's the name there for you. And the choroid, especially at the back here, the choroid is very dark. It's almost black. It's a purplish black color. And it absorbs light very, very easily. And its purpose is to absorb light which has passed through the retina, so that we don't have light reflected around the inside of the eyeball. Instead, light is absorbed at the back by the choroid. The choroid is this thin tissue, but at the front here, the choroid is transformed into these muscular extensions here. And there are two kinds of muscles here that we can see. The first is, remember that this is a section through the eye. So if we were looking from the front, it would be a band of muscles like that. There is a band of muscles all running all the way around called the ciliary muscles. And here they are here. We'll discuss their function in a minute. The next lot of muscles are in this part of the eye called the iris, which is obvious to all of us. That the iris is what gives your eyes their color. And you'll no doubt be perfectly well aware that the iris can open and close. And the iris opens and closes in response to two stimuli. The one is brightness of light. If the light level is very low, then the iris will open to admit more light into the eye for image formation, for clear image formation. But there's another circumstance, and that is if you look at something very close by, if you try and focus on something very close to your eye, then the iris will close to almost like a pinhole. And that is because an opening like this, like the iris, actually functions in many ways like a lens. Because the edges of the hole interfere with light waves passing through, it actually focuses light to a certain extent. Much more, it works much better if the hole is very small. You can do this yourself. You can take a piece of paper and you can punch a hole in it. Punch a hole and hold it up against, close to a wall with a light behind it. You can actually cast an image of the light onto the wall. That hole in a piece of paper can act as a lens. And this is much the same here with the iris. If we pull the iris closed, then it has a focusing ability. So we need to remember that also when we talk about overall focusing in the eye. The iris is closed with sphincter muscles. Those are muscles which are joined to one another. All the sphincters in the body, the muscles are joined to one another to form almost circular band which can be closed. And then the iris also has muscles which can pull it open. It has muscles on the side which pull it open like a curtain being drawn, be closed by the sphincter muscles and opened with these lateral muscles. So remember, the iris has a hole in the middle called the pupil, and the pupil can be closed in response to very bright light or an attempt to focus close by on something. So behind the iris, there is a clear crystalline body here called the lens. And the lens is made of specialized proteins. And the lens actually is elastic. Its normal shape is round. But these is tied here on the side to these muscles here. And we'll see it again in a minute. So that these muscles, the ciliary muscles, can act on the lens. We'll see that in a minute, exactly how it does it. So those are our two layers, sclera, then the choroid, and then forming all of this here. The third layer is the retina. And the retina is a paper-thin layer right on the back of the eye. It's actually not even very tightly tied to the back of the eye. It's held, it is fairly loosely held, which has important consequences. especially later on in life, it can actually buckle and tear away from the back of the eye. But normally it's very, very closely applied to the back of the eye here by pressure inside this jelly-like material here called the vitreous body. There's a slight pressure which presses the retina flat against the back of the eye. The retina we'll see its structure. It's actually really complex. Very, very thin. That is our light perception. That is where all of our light perception takes place. And the information, the stimulation of those cells, the nerve impulses are sent via the optic nerve here to the brain. And we'll see that exactly what happens in a minute. So make careful note of all of the anatomy here. On the outside here, there are called the extrinsic muscles. There are 3 pairs of them. You don't need to know the names. Just realize that we have 6 muscles which can rotate the eyeball in all directions. And here, this is the structure of not only the lens, but the whole of the front part of the eye, which is the image forming part of the eye. First of all, here is the cornea, the clear cornea. And the cornea is tremendously important in focusing. Most of the focusing of the image is actually done by the cornea. And the rest of the eye actually refines the image which the cornea makes. Behind the cornea, here's the iris, here, with the pupil through which light is going to. And remember that the iris can actually form a focusing ability as well. If you pull it closed, you pull the pupil closed, then it's amplified, the ability to form the image. So the pupil can also be used in refining the image initially made by the cornea. But the most important refining mechanism is here in the lens. The lens is absolutely, under normal circumstances, the lens is absolutely clear and elastic. Its natural shape is fairly round, and it's on the side here. You have to look carefully on this diagram. But on the side here, there are ligaments, which attach the lens to these muscles here. These are the ciliary muscles here, forming a band underneath the iris. And those ciliary muscles can contract, and they will pull the lens flat. Then when the ciliary muscles relax, the lens is elastic, so it naturally just rebounds to its round shape. And in this way, by pulling the lens and letting it round, pulling around, the focus of the image can be refined. And the idea is that it be refined so that it falls exactly onto the back of the eye. It forms exactly here. That is the ideal position for image formation. For some reason, the lens, for example, is too round, the image will be formed in front here. If the lens is too flat, it'll fall somewhere behind here. And in either of those circumstances, the image would appear blurred as it falls. You'll still see an image, but it won't be in sharp focus. Initial image is made by the cornea, is formed by the cornea, refined by the iris, if need be, and then absolutely refined by the lens to fall exactly here. And you see this point here, right opposite the lens. That is called the fovea. And the fovea is the point at which the sharpest focus is going to fall. So the image, that's right opposite the lens there. So when we look, when you go have your eyes examined, the ophthalmologist will look through the lenses at your eye and the back, shine a light on the back of your eye, etc., etc. This is what they see. And it's a very revealing picture for many health conditions, because the retina and the structure of the eye is one of our organs that does get affected by many different kinds of disease, and different symptoms may appear in the eye, in particular things like diabetes, just aging, high blood pressure. All of these things can have an impact on the structure that an ophthalmologist will observe at the back of the eye. So as we look through here, the first thing we notice, that little dot there, that little bump. That is the fovea. That is where it's the very center of the image that is going to be cast on the back of the eye. If you look around it, you'll see here, there's a yellowish area. And that is called the macula. M-A-C-U-L-A, the macula. Sometimes you'll see it also called the macula lutea. Lutea simply means yellow, and it's because of this yellowish pigmentation. This is a very important area, because this is where our retina is richest in light perception cells. They are very densely packed here. Not only that, here in the macula, we have 4 kinds of light perception cells that I'll show you in a minute. And this is where all our color perception takes place, is there. Now you can imagine, if you can do this, just look straight ahead of you. If you can, look at something where there's color, look at the picture on the wall or whatever. Keep your eyes steady, and that image that you see is being cast. The center of that image, right in front of you, is being cast here. And you will perceive it in complete normal color. You see all the colors in it. But just look very, very carefully. Don't look. Just be perceptive about the periphery, about your peripheral vision. Your peripheral vision is where the image is being cast, is here on the outside of the macula. And there are very few color perceiving cells there. And in fact, if you are able to perceive it really carefully, you will realize that in your peripheral vision, you see it mostly in terms of black and white. So in most of the image that you perceive over here is in black and white. The next thing you notice is that this is richly invested with blood vessels. But not only blood vessels, there are many, many nerves running through here as well. And all the blood vessels and the nerves, they converge here, at this point here. And this is where the optic nerve is leaving the eyeball. This is also called the blind spot. It's called the blind spot because it does not have any light perception cells there. And you remember in the first short class that we did, I pointed you to a little experiment where you can take 2 images, hold them away from you, see both of them, move them closer and closer and closer. And eventually one of the images will be cast on this. So it was a plus sign and something. And in the end, the plus sign gets cast on here, and bingo, it disappears. And that is because the image is being cast onto, when you bring it close enough, it gets cast onto the blind spot where the optic nerve is leaving the eyeball. So I'll just tell you a couple of other things about it. One of the unfortunate consequences of aging can sometimes be that perception is lost in this area of the macula. And there are 2 conditions. One is where the macula simply thins and cells begin to die out and get lost from the macula, and all light perception gets lost, and people then start to perceive a darkening right in the center of their vision. The other kind that is called macular degeneration. And when it's from the tissue thinning like that, it's called dry macular degeneration. But there's another form called wet macular degeneration. That's when thousands of blood vessels begin to encroach. You can see the blood vessels here are mostly very, very fine, very small, and not that common right around the fovea. But in wet macular degeneration, these blood vessels start to proliferate and they interfere with the functioning of the macula, interfere with image formation. And again, it starts to darken and lose perceptive ability. The other common condition, especially currently in America and in the Western world, is the impacts of diabetes. And diabetes leads to a retinal degeneration of the retinal tissue and can result in complete blindness, certainly to loss of visual acuity. So let's have a look and see actually how the image is formed and what happens to it. Well, the image that is formed is what we call a real image. And a real image is completely inverted. It is completely inverted, and left and right are reversed. So there's a complete reversal of the image that falls on the back of the eye here. Again, the image is formed from its interaction of three things: the cornea, the iris, and then the lens. The lens can be, as we've said, can be rounder, flattened to produce exact positioning on the retina itself at the back here, unless there is something wrong, and that we'll talk about in a while. So the image that is formed there results in nerve impulses, in a set of nerve impulses. But here's the thing to think about. You're not actually making an image on your eye. You're stimulating nerve endings. You're causing nerves in the eye to actually send messages to the brain. And the brain has to interpret those messages. And once it's interpreted them, it has to refer that back so that it appears to us, it looks like there is an image in front of us. That image in front of you is actually in your brain. It is being created in your brain by the nerve impulses landing on the retina. So it's a complex process. And most of that visual processing is done here at the back of the brain, on the thin layers of nerve endings here on the occipital lobe, at the back. And this is called the visual cortex. It's the thin layer on the outside of the cerebral hemispheres at the very back of the skull. So it reaches the visual cortex by passing from the eyes, the optic nerves, the impulses, etc., pass through the thalamus. You remember, the thalamus is part of that primitive part of the brain. And for many of those areas, their principal responsibility is directing nerve impulse, the shunting nerve impulse. And the initial shunting through the thalamus takes visual information and ensures that it goes to the visual cortex. It ensures that it goes to the visual cortex, but remember something. The thalamus is also receiving all sorts of other information. At the same time as you are seeing, you are hearing. And that information is also being shunted through the thalamus, and the thalamus has to decide, this is hearing information, send this off to the temporal lobe. That's the first way station for hearing. This is visual information, send this to the occipital lobe at the back, etc. There are some circumstances where these things can get confused, where the thalamus can make mistakes and start sending stuff to the wrong place. And one of these is if you take hallucinogens of one sort or another. This is one of the things which hallucinogens do. And they interfere with the thalamus' ability to decide what belongs where. People start seeing colors where they are hearing something. They start hearing colors. They see color and they hear things. All sorts of other sensory input gets misinterpreted because it gets shunted to the wrong places. And don't forget that this visual image that is created in the occipital lobes, we're still got to be interpreted. You've still got to know that you're looking at a television, that you're looking at a computer, that you're looking at a book. All of this information is then sent from the visual cortex. The image information must be sent out to other areas of the brain, which enable you to recognize what you're looking at. If you look at a face, this part here just recreates that face that you're looking at. But the recognition of that face actually takes place down here in the front of the brain, in the frontal lobes. So this has to communicate with the frontal lobes. It is a very complex process. So it is quite possible, for example, that some conditions such as schizophrenia, that sort of thing, at least part of what is happening is that the brain has lost its ability to distinguish between these different sensory inputs. People hear voices where there is no sensory input. That sort of, they see things, they have visions, whatever, where there is no stimulation of the retina. These are all errors being made in these different parts of the brain. Okay, so here's another thing to think about. We actually have two eyeballs, and under ideal conditions, they coordinate with one another and with the brain. But let's have a look, and you realize that we have a left eye and we have a right eye. But the information that is perceived on your right is actually analyzed on the left, in the left-hand side of the visual cortex. What happens, what you perceive on your right, on your left is analyzed on the right-hand side of the visual cortex. So the wiring is actually quite complicated. Have a look here and you'll see. Let's just take what we see on our right-hand side, okay? So first of all, your right eyeball, here it is. Don't forget, the image is completely reversed. So here it is, what is on the right is here on the left of the eyeball, and that is sent off here to the left-hand side of the brain. So what is on the right, perceived on the left of the eyeball, and is sent off here to the left-hand side of the brain. Same, what is perceived on the left is sent off here to the right-hand side of the brain. Same thing here. So you see what is happening. We have a crossover. The optic nerves actually cross their apothalamus. And it's a place called the optic chiasm. That simply means a cross. It's the optic cross. The optic chiasm is where these nerves actually cross over to shunt the information to the correct side. So that has important implications. First is, in actual fact, what the right-hand eyeball sees is different to what the left-hand eyeball sees. And you can tell that by closing one eye, by blinking from one eye to the other, keeping your eyeballs absolutely still. You'll see that the image shifts like that, because what you see in one eyeball is not necessarily what you see with the other eyeball. And those two images have to be reconciled. And they are reconciled by the two sides of the visual cortex talking to one another and in analyzing the image they are receiving, the visual cortex actually, first of all, it inverts the whole image. That's its first responsibility. I told you what falls on the back of your eye and what is transmitted to your visual cortex is an image which is completely inverted and flipped around. That's not going to be very useful for us if we walk around in a world which is completely upside down visually. So the visual cortex actually puts it all into the correct order. And this is very easy to demonstrate because you can, in fact, get people to wear glasses with lenses in them, which do the opposite. They will invert. When you look through them, the world is inverted to you. So then, if that is the case, then the image is falling correctly on that. I mean, upside, the right side up, right, on the eyeball. But what you perceive is all upside down for a while. After a couple of days, your brain actually, the person who's wearing those inverted glasses, your brain actually learns to turn everything upside down again and put it into the right. Leave them, okay, let them, now they're quite happy. Now they're wandering around the funky glasses, but the world again will look normal to them until they take the glasses off. Now your brain is totally flummoxed because now its correction is incorrect. Now you take those inverted glasses off and you're seeing as you normally do, but the brain still thinks it's inverted for a while. Then it gets used to it and it puts it back again. These things are not hardwired into us. They are actually, the visual cortex can actually be trained to do things, to invert the images, etc., etc. Okay, so I'm only going to do this in relatively simple terms, so don't freak out when you see this complicated diagram. But this is the structure of the retina. It's several layers of cells. First of all, remember, we're talking about special senses. And the special senses are those where we have a sensory cell which is specially adapted to produce a nerve impulse when it is stimulated by something in particular. In the case of the eye, we have cells which are specially structured to produce a nerve impulse when they are stimulated by light. And we have 4 of those cells. The first of these, they're called the rods. And you can see they're here. And the rods perceive only in terms of black and white. So they see gray, but the only color choice they have is between black and white and mixtures of it. Those are the rods. But these here in between are the cones. And there are 3 kinds of cone. There were those which perceive green, those which perceive red, and those which perceive blue. And they have an optimum wavelength that they will respond to, but they will respond to a range of wavelengths in that kind of color range. The greenish color, the reddish, and then the bluish colors will all stimulate them. So this now raises the question, well, we don't only see in terms of red, green, and blue. We see all sorts of other colors. We sort of see a whole range of colors. Well, how is that achieved? And the answer is, the easiest demonstration is to think, what is the one important color which is missing that we don't have a cell for? We've got red, green, and blue. We're missing yellow. We have no, and yet we can see yellow perfectly easily. We see a wide range of yellows in actual fact. And the answer is, the yellow will stimulate both red and green. It will stimulate both of these cones. And that is interpreted, if both of them are stimulated, the brain interprets it as yellow. So I can produce a yellow sensation by two means. I can either shine yellow light on the retina. Sorry, I can do it this way. I can shine yellow light on the retina, stimulate both of these, and produce yellow. It'll send a message yellow to, it'll send messages to the brain. The brain will interpret it as yellow. Or else I can shine green light and red light equally onto the back of the eye. And guess what? It'll also look like it's yellow. The same thing will happen. The brain will interpret that as yellow. That's very useful. It's very useful in one particular circumstance. For example, and that is, if you look at your television screen, if you go up really, really close to your television screen, you'll see that the image is made up of little tiny dots of color, but they're only green, red, and blue on your television screen. They use the same system. They are, when you see yellow on your television screen, they're not actually broadcasting yellow light at you. They're broadcasting green and red light equally at you. And your brain interprets that as yellow. So all of these intermediate colors that we see, the yellow, and then all the oranges, all the different colors of blue, all of these things, the range of color is produced by interaction between green, red, and blue cones sending their messages to the brain, and then the brain interprets that as color. I will tell you one very interesting thing, and that is the question has been asked why, that we know that some animals have more cones than we do. They perceive a wider range. Some simple animals, for example, have cones which can perceive down into the ultraviolet, which we can't see at all. And others can see into the infrared. They can see long wavelengths of light that we cannot see, we're unable to perceive. So the question has been asked in evolutionary terms, why is it that our range is somewhat restricted? We can still see a lot of color, but is somewhat restricted compared to others. It may have something to do with our evolutionary origins in fairly dim light, in forests, in dense forest, and this sort of thing. But I will tell you one very interesting thing, and that is by looking at a huge range of populations, of people in different populations, they have discovered a few people who do have another cone. I don't know exactly how it works, but I think that it is in the yellow range. They have a cone in the yellow range, and their color perception is enormously greater. I don't think that that's a huge evolutionary advantage, but it's actually extremely rare in human beings. For the rest of us, we're stuck with green, red, and blue, unless, of course, you are colorblind. And this is the origin of color blindness, the commonest color blindness. One of these cones or more one of these kinds of cones is unable to respond correctly to the wavelength that you're supposed to respond to. So, for example, the commonest form of color blindness is red-green color blindness, where one of these fails to operate properly, and that person cannot distinguish between red and green. May also have implications for their perception for other colors, such as the yellows. And this is a heritable characteristic. Okay, so there are all of our light perception cells there, rods, and then the three kinds of cone. Notice behind here, this is actually the choroid lying behind here, the black or purplish black layer that absorbs all the light. Now, wait a minute, this looks strange. Surely this is upside down. Well, it's not. In actual fact, the light comes in this direction here. One would think it would come in this way, right? Straight onto the light perceiving cells, but it doesn't. The light perceiving cells actually lie at the back of the retina, not at the front. Here, don't worry about the complexity of it. Here are the light perceiving cells, rods and cones, and they are communicating with nerves, but through a complex set of interlinking nerves to a whole network of nerves here, which will now run off and go off to the optic nerve to send the message to the brain. The choroid, remember, is at the back. The light is coming through this way, like this. And the choroid absorbs that light once it's passed through the retina, because the back of the eye, of course, is white. And if the light hit the white light, it would bounce back here and it would start stimulating these again, which is very undesirable. You want the light to only be one way. The image to be cast there and then absorbed by the choroid. Okay, so this just shows you exactly how it's organized. Here's a little patch of retina here in the macula. So we've got lots of cones in it. There are the rods and cones there, and you can see the light is passing through the nerves at the front to the light perceiving cells at the back and is absorbed here by the choroid. Now I'll just show you here. This is the actual structure. So the rods and the cones actually have different structures, which you don't need to worry about. But I just want to point one thing out. You see here on the rods not only perceive black and white, they are also able to perceive very dim light. Normally, under norms of daylight and everything, they just function, have a baseline perception and everything. But have a look here. You see this like piled up stuff. This is a pigment. It's specialized pigment for rods, and it is called rhodopsin. And under normal circumstances in daylight, there's not very much rhodopsin. It doesn't need it because the light is bright, and it's the rhodopsin, it's pigment that actually is responsible for creating the nerve impulse ultimately. But when you go into the dark, there's very little rhodopsin, and for a while, your rods are completely unable to perceive light. Go into dim light from bright light, and you're completely blinded. Right? Everything is pitch black. But then, if you just stand for a while, with your eyes open, slowly you start to perceive the image growing and growing and growing in front of you. And what is happening is that the rods are actually manufacturing rhodopsin. So they manufacture rhodopsin to the point that they're actually able to start firing. And you start to be able to see in dim light. Also, if you're in dim light, you've been there for a long time, and you walk out into the bright light, you're absolutely blinded for a minute or two. And that is because your rods are actually packed with rhodopsin, and they go into bright light, they send blindingly strong signals to the brain, way too strong. And you react by closing your eyes until you've broken down some of the rhodopsin. You reduce the sensitivity of the rods, and then you can see normally once again. Okay, I've explained to you how you see. And there, if you just move on, then you'll see that there is a movie about color. And watch both of these. This is a long movie, just if you're interested in color. And maybe if you're an artist or whatever, maybe this would be particularly interesting for you, about the biology of color. But it's a long movie. You don't need it. These are short and just explanatory. Okay, so let's just spend a little while talking about some of the diseases and disorders of the eye. And the first one I mentioned, because this is another disease which unfortunately seems to be becoming more common, maybe due to various things like lifestyle, diet, sort of things in the United States and in Western world. And this is the condition called glaucoma. And glaucoma, I'm just going to flip back all the way back to here. Glaucoma, I just need to remind you, behind the eye here is that body called the vitreous humor. It's a gel-like substance, and it does not circulate very much. It doesn't change very much, even. But in the front here, there is a fluid called the aqueous humor, and that is continually in circulation. There are canals that you can just see here. You see this little tiny dot there. There are canals which absorb that fluid called the aqueous humor, and it is continually produced and circulates through the this front of the eye here. But in glaucoma, these canals, the whole drainage system for that aqueous humor is disordered, and you begin to build up pressure inside the eye, and especially the pressure is transmitted to the retina through this through the vitreous humor, because fluid is basically incompressible, right? So if I'm building up fluid in the front of the eye, the pressure gets transmitted throughout the whole eye, and that is extremely damaging to the retina. Glaucoma is, throughout the world, glaucoma is a major cause of blindness. There are various reasons that can happen. There are disease processes. Aging contributes to it. It is racially determined as well. Here in the United States, much more common amongst African Americans. And in Africa, extremely common. In third world countries, there are many different contributing factors to causing of glaucoma, including things like parasites and things like that. So when you go to the ophthalmologist, as you should do, you should go for a yearly checkup of your eyes. One of the first things that they'll do is they test the pressure inside your eyes, usually just by blowing a puff of air onto the cornea and measuring how it reacts, how it bounces back. Glaucoma is eminently treatable by drug therapy, but also by surgery if need be. The next disease is one which occurs to one extent or another in virtually all human beings as they age. And this is the condition having cataracts. And cataracts is clouding of the lens, and it's simply a process of aging of the proteins that make up the lens. And it is largely due to ultraviolet light damage of the lens. And so the lens begins to cloud. Again, it is eminently treatable. It is tolerated to a large extent. Many people never actually ever realize that they have some degree of cataracts. But if it becomes severe, the lens can actually be removed and can be replaced with a synthetic lens, which works almost as well. And I should also say, if you do lose the lens entirely and it's not replaced, you can actually form a pretty good image anyway on the back of the eye, just by the cornea, etc. Macular degeneration, I've already told you about. It's degeneration of that macular tissue at the back of the eye. Here's one which is probably virtually everybody has had at one time, incredibly irritating and annoying. It's a bacterial infection, either of the follicles of the eyelash, that's the commonest, or sometimes infection. I don't know if you remember, but I told you that in the eyelid, there are little glands that produce lipid, which is part of the tears. It's an essential part of tears. And those can also become infected. and it forms like a little abscess. And this is staphylococcal infection is the commonest. Staphylococcus is the commonest cause of this. Very, very, very painful and annoying. And often it is simply treated symptomatically by hot compresses, that sort of thing, to try and get the abscess to open and drain. But I mention it because if you are a person that has persistent styes, you might want to have it checked, because what does happen sometimes is that the bacteria set up a population in your sinuses, and that communicates with your eye, and you've got a continual source of infection, and in that case they can be easily treated with antibiotics. Dry eye, I think I've already told you everything you need to know about dry eye. If you listen to the very first little lecture. But the dry eye, important, because we're all tending to subject ourselves to dry eye syndrome because of something to do with screens. Screens induce us to keep our eyes open for too long, and our eyes actually physically dry out. But dry eye has other consequences. When you blink, you produce tears. That is the normal situation. Don't blink, your eyes will tend to dry out. Okay, I'm going to stop there and discuss the other disorders of sight at our next lecture, when I will also discuss the ear. And so for the moment, we'll stop there.